What if you had like a molecular design studio right at your fingertips? Imagine you could just build the basic blocks of life proteins for all kinds of amazing things, new medicines, better farming.
Right.
The really big question is, could we actually teach a computer the blueprint of life well enough that it could design these proteins for us when we need them?
That's exactly the exciting frontier we're looking at today. We're diving deep into some really cutting-edge research, specifically focused on a paper introducing something called ProGen3.
ProGen3.
Yeah, it's a new family of protein language models.
Protein language model. So it sounds like we're literally teaching AI to speak protein.
Yeah.
What's the goal here with ProGen3? What are they trying to achieve?
Precisely. Our plan today is to really unpack what these ProGen3 models are, how the scientists actually built them, and maybe most importantly, what they can do. Think of it as getting you up to speed on a pretty major leap forward in how we might design, well, the very stuff of life.
Okay, and the research paper we're digging into is titled 2025.04.151.649055v1.full.pdf for anyone wanting the source. And for you listening, we know you want the key stuff without getting totally bogged down in jargon.
Yeah, it can get technical fast.
So we'll be your guides trying to pull out the most exciting bits from this research.
Sounds good. So let's start right at the beginning. What are protein language models?
Well, my understanding is it's kind of similar to how computers learn human language, right? Like GPT and models like that.
Exactly.
These models learn the patterns, the sort of grammar of proteins by looking at, just billions of naturally occurring protein sequences. It's like the AI is reading the instruction manual of life.
That's a great analogy. Just like a language model learns grammar and word relationships from text, a protein language model learns the connections between amino acids. Those are the protein building blocks. By studying, billions of sequences. The main goal is to understand the complex patterns, the distribution of proteins found in nature.
And then use that knowledge.
And then use that understanding to actually design new functional proteins. And this paper, importantly, includes experiments showing they can design novel proteins that work in the lab for different uses.
Okay, so ProGen3 is the latest step here. What makes it, special or more advanced?
Well, ProGen3 is described as a family of sparse generative PLMs. Let's break that down. PLM is protein language model, like we said.
Got it.
Generative is key. It means they can create new sequences, not just analyze old ones. And sparse is interesting. It basically means the model doesn't use all its computational brain power all the time.
So it's more efficient.
Exactly. More efficient. And these things are massive. The biggest one has up to 46 billion parameters.
Wow.
And it was pre-trained on this huge data set, 1.5 trillion amino acid tokens. Just the sheer scale is a big part of why they're so capable.
46 billion parameters.
Yeah.
That gives you a sense of the complexity. Now, the paper mentioned something called the Profluent Protein Atlas V1, or PPA1. What's that?
Right, the PPA1. That's the specific data set they curated to train ProGen3. It contains 3.4 billion full-length protein sequences.
Full length. Why is that distinction important?
Well, they deliberately excluded protein fragments. The idea was to make sure the model learned from complete protein examples, understanding the whole structure from start to finish, not just bits and pieces.
Okay, that makes sense. And they didn't just dump all this data in, right? You mentioned they optimized how the data was presented.
Exactly. They didn't just use the raw natural distribution of proteins. They actually experimented with different ways to balance the diversity of protein families in the training data.
How so?
They tried things like giving all families equal representation. They called that uniform. Then other methods, square root and inverse log, which tend to give a bit more weight to less common protein families, and they compared these to just using the unmodified natural frequencies.
Trying to find the best way to teach the model about the whole protein universe, basically.
Precisely. And they found that the inverse log distribution worked best.
Best in what way?
It gave the lowest loss, which is like the model's error rate, especially for sequences that were very different from the training data, what they call out of distribution. So slightly emphasizing the rarer proteins helped the model generalize better to truly novel stuff.
That's really interesting. And you said the uniform approach treating everything equally actually did the worst.
Yeah, that was quite telling. It suggests that the natural frequency of proteins, even the very common ones appearing many times, actually contains important information for the model. Just flattening everything out lost some of that signal.
So nature's redundancy isn't just noise, it means something.
It seems so, though as the inverse log result shows, maybe a little bit of rebalancing can help for certain goals, like generalization.
Right. Okay, let's talk more about that huge 46 billion parameter model. The paper mentions compute optimal scaling laws. What are those about?
Think of it like finding the sweet spot when you're training these giant AI models. Scaling laws help you figure out the best balance between how big your model is, parameters, and how much data you train it on, given you only have so much computing power.
Getting the most bang for your computational buck.
Pretty much, yeah. How to spend your compute budget wisely for the best learning.
And how did the 46B model actually do compared to what these laws predicted?
Well, here's a cool bit. It actually did better than predicted. It achieved a lower validation loss, meaning it learned more effectively than the scaling law suggested it should for that amount of compute.
Oh, nice. Any idea why it overperformed?
They hypothesized that it might be related to the warm-up period during training. They used a longer period where they gradually increased the learning rate at the start.
Okay.
And this longer, slower start might have helped stabilize the whole learning process for such a massive model, allowing it to reach a better final state, like easing into the learning process.
Interesting. So you've got this huge, well-trained, possibly even overperforming model. What happens when you ask it to generate new proteins? How does size impact the output?
Right. They did a lot of computational analysis on this. They compared 3 sizes, a 339 million parameter one, a 3 billion, and the big 46 billion one.
Yeah.
First off, the larger models were just much better at generating sequences that actually look and behave like real proteins computationally. They passed more quality filters, not too repetitive, had the right stop signals, aligned well to known natural proteins.
That seems intuitive. Bigger model, better quality. What about the variety of proteins?
Yeah, this is where the scale really shines. The larger models could generate proteins from a vastly wider range of protein families. They covered much more of the potential protein sequence space.
So they weren't just better, they were broader.
Much broader. In fact, almost everything the smaller models could generate was basically a subset of what the larger models could do. The big models could even generate sequences from families that the smaller models found, like totally unlikely or perplexing.
Huh.
And importantly, they were also way better at generating sequences from those smaller, less common protein families. So size really unlocked a much greater diversity and design potential.
Okay, so computationally, bigger seems better, especially for diversity. But, a computer saying, this looks like a good protein isn't the same as it actually working in a cell, right? This is where the lab work comes in.
Exactly right. You absolutely need the wet lab validation. They use a technique called a split GFPE coli protein expression assay. It's basically a test to see if the generated proteins can actually be produced inside bacteria in a stable, soluble form.
And what did they find when they tested these AI-designed proteins in the lab? They looked at proteins from different sized models, right?
Yeah.
And ones that were more or less similar to known proteins.
They did. And the results were a bit surprising, actually. All the models, regardless of size, 339M, 3B, 46B, they all achieved pretty similar rates of successful protein expression in the E. coli.
Wait, similar rates? So the huge model wasn't necessarily better at making individual proteins that worked?
Not in terms of the overall hit rate, no. But, and this is the crucial part, the larger models achieved those similar success rates across a much, much wider diversity of protein families.
Ah, I see. So the 46B model could successfully design working proteins from families the smaller models couldn't even touch.
Precisely. The breadth of what they could successfully design and express was vastly larger for the bigger models. And interestingly, the expression levels of these generated proteins were generally comparable to randomly chosen natural proteins from the same families.
So they weren't just working, they were working kind of like natural ones. It's pretty good.
Yeah, it shows the designs are biologically plausible, at least in terms of expression.
What about going totally off-piste? Designing proteins really different from anything known? Like less than 30% similarity to anything in the databases.
They pushed that boundary, too. They specifically generated sequences with that low similarity. And remarkably, all the models, even the smaller ones, could generate these highly novel proteins that still expressed in the lab. But here's another fascinating detail. In that really novel sequence space, The proteins generated by the larger models tended to show higher levels of expression.
Okay, hold on. So when designing things really new, the bigger models not only could do it, but the results were potentially better, more robustly expressed.
That's what the data suggests, yes. It hints that the larger models aren't just better at mimicking known nature, they might actually be better explorers of entirely new functional sequence space.
Right, it's wow. Okay. The paper also talks about protein infilling. What's that?
Right, infilling. So ProGen3 isn't just about generating whole proteins from scratch. It can also redesign specific bits within an existing protein.
It's like editing a specific part of a protein sequence.
Exactly. Imagine you have a protein and you want to change a loop or a segment to maybe improve its function or stability. ProGen3 can take the surrounding context and propose a new sequence for just that section you want to change.
And they tested this on what kinds of proteins?
Yep. They tried it on therapeutically relevant proteins, industrially useful ones, and also just random proteins from E. coli. They tested filling in both short gaps and longer segments.
And how did the different models compare on this editing task?
The 46 billion parameter model generally performs slightly better than the 3 billion one, especially when infilling longer segments. It suggests the bigger model has a slightly better grasp of the overall context of the protein when making those edits.
Okay, slightly better. Any standout results?
Yeah, actually, some of the sequences ProGen3 generated through infilling actually showed improved expression levels compared to the original natural protein they started with.
Well, so it didn't just replace the segment, it potentially made the protein better in some way, like more stable or easier to produce.
The improved expression suggests something like that, yeah. It opens the door to taking existing useful proteins and using AI to potentially enhance them.
That's huge. Okay, the last big section seems to be about aligning models with laboratory data. This sounds important. What's the deal here?
It really is important. So you have these massive models trained on sequence data. A common task people try is zero-shot fitness prediction. Basically asking the model without any extra training how a mutation might affect a protein's function.
Okay.
Now, here's a counterintuitive finding. They discovered that once models got really big, like beyond 3 billion parameters, they sometimes got worse in the specific zero-shot prediction task.
Worse, but they're bigger and supposedly smarter.
It seems that as the model gets incredibly good at presenting the sheer diversity of all proteins, it might lose some focus on predicting the very specific subtle effects of single mutations within one particular protein. It's like becoming fluent in all languages, but maybe slightly less precise in the grammar of one specific dialect.
Huh. So a trade-off between breadth and that specific kind of depth.
Kind of, yeah. But, and this is key, they found you can unlock the power of those huge models by aligning them. They used a method called IRPO, Iterative Reasoning Preference Optimization.
Okay, what does that involve? More training.
It involves fine-tuning the model using relatively small amounts of actual lab data. You give the model some real-world experimental results to help guide its predictions and generation.
Like giving it a dose of reality.
Exactly. And the results were dramatic. For example, they aligned models using data on just a few mutations in a protein, what they call local mutational landscapes.
Right.
After this alignment, the larger models became incredibly good at predicting the fitness effects of mutations much further away, involving many more changes than they were explicitly trained on. The aligned ProGen3 46B performed as well as, or even better than, specialized tools built just for that task.
Wow. So a little bit of targeted lab data dramatically boosted its predictive power.
Massively. They saw similar success when aligning with protein stability data. The aligned larger models got much better at predicting stability, especially for very different proteins or those with multiple mutations.
And did this alignment carry over into designing better proteins too?
Yes. When they used the stability-aligned models to generate new sequences, those sequences were predicted computationally to be more stable, and crucially, when tested in the lab, this stability alignment often resulted in generated proteins that had significantly higher expression levels.
Got it. So the alignment didn't just make the model a better predictor, it made it a better designer of proteins that actually work well in the lab.
Precisely. It bridges that gap between the vast sequence knowledge and the practical realities of experimental biology.
So, summing up the bigger picture here, what are the main implications of this ProGen3 research?
Well, it really drives home that these large-scale generative protein language models, especially when trained on well-curated datasets like PPA1, are becoming incredibly powerful tools for all sorts of protein design tasks.
And the scale matters. The sparsity helps efficiency.
Right. And as we keep scaling them up and finding smarter ways to train and apply them, like this alignment technique, their capabilities are just going to grow. ProGen 3 46B feels like a really significant milestone towards truly bespoke protein design for new drugs, custom enzymes, novel materials.
The possibilities seem enormous. Of course, with designing life's building blocks comes responsibility. Did the paper touch on safety or ethics?
They briefly mention working within safety guidelines and mention the International Gene Synthesis Consortium, which provides screening protocols. It's definitely a background consideration that becomes more important as the technology gets more powerful.
Right, absolutely. Okay, so just to recap the key takeaways for everyone listening.
Yeah.
You've now got a handle on ProGen 3. You know, it's a huge scale, and that optimized training let it generate a much wider variety of potential proteins, even stuff unlike anything seen in nature.
And critically, how aligning these models with real lab data makes them much better predictors and designers of proteins with desired properties, like stability or high expression.
The real aha moment, I think, is just grasping that we can use AI, trained literally on the language of biology, to start designing our own biological tools.
It's a fundamental shift, really, from observing and modifying nature to actually designing biological components with intention.
So here's something to think about.
Yeah.
If we get really good at designing specific proteins on demand, what happens next? What kinds of completely new medicines could we create? What about self-healing materials or enzymes that break down plastic waste?
Yeah, the potential applications are vast. And alongside that, what are the ethical guardrails we need to build as this tech develops? This ProGen 3 work really opens up a huge landscape for future research and innovation. You basically just had a primer on the cutting edge.